This is a lecture for Chapter 25 on meiosis and inheritance. Remember that there are two types of cell reproduction. There's asexual reproduction. This is when one parent cell gives rise to two identical daughter cells. Some types of plants do this, some types of ivy. But for this process, mitosis is used, which we've already talked about in Chapter 24. So just making another cell using the one that's already there. The other type of reproduction is sexual reproduction, and this involves having two parent cells. And that can happen in any number of ways. So obviously with animals, they go through some sort of act of exchanging those parent cells, whether it's mammals and they're actually engaging in sex, or it's fish and the female lays her eggs and the male swims over the top and the sperm fertilized the eggs. Lots of different ways for creatures to make that happen. Even in plants, the pollen and the egg cell in the plant, that's considered sexual reproduction as well. So in that, the key here is two different parents, and by combining their genetic information to make one new unique daughter cell. And for this, we use meiosis. Gametes are the cells from these parents that fuse together to form their offspring. So in humans, the female gametes are eggs. They're really big. They're the largest of our cells in the human body. They're non-modal, so they don't move on their own. That said, they do travel down the oviduct, but they don't have a flagella or anything like that to help them swim. The male gametes are sperm. They are much smaller, so you can see from our picture over here, here's a sperm cell, and this big one is the egg cell. Very, very different structures for these two gametes in humans. And they are modal, so their tails are a flagella. They actually spin to help them move. Gametogenesis, which is the process of actually making these gametes, the eggs or the sperm, occurs in gonads. So in females, those would be the ovaries. In males, those would be the testes. Chromosome number is something important for us to take into consideration of these, because if you're putting two cells together, you want to make sure that the combination of those two has the proper number of chromosomes. So the number of chromosomes in an egg or a sperm is actually half the number that you would have in any of your other cells. Meiosis is the process that actually reduces that chromosome number from diploid, which is 2 copies, to haploid, which is 1 copy of each chromosome. That way, if you have 1 copy of each chromosome in a sperm and 1 copy of each chromosome in an egg, once you put them together, now you have 2 copies of each chromosome to make that embryo, to make that baby. This only happens, so meiosis only occurs in ovaries and in testes. Nowhere else in the body will meiosis happen. Everywhere else in the body uses mitosis to make more cells. Only the ovaries and testes use meiosis. So here we have just a little diagram to help you understand how these chromosomes are being divided. So here on the left are just a description of chromosome 1. So in all of your cells, you have 2 copies of chromosome 1. You have 1 from your dad, let's say that's the blue one, 1 from your mom, we'll say that's the red one, and you would be considered diploid. You've got 2 copies of chromosome 1 there. As you go through the cell cycle, DNA replication happens, and now you've made an identical copy. So now here with your blue original copy, you've got the identical copy right next to it. They're being held together here at the centromere in the middle. Same thing with the red copy, you've got the sister chromatid right next to it. And those are those X shapes that we're used to seeing. So the identical copies are your sister chromatids. The different types that you have, the ones from dad, ones from mom, those are homologous chromosomes. So in humans, you have 23 different types. Those are numbered 1 through 22, and then the X or the Y chromosomes. And you have 2 copies of each one of those. So 23 types, so chromosome 1 being one of those types. And you have 2 copies of each one of them, giving you a total of 46 chromosomes in each one of your cells. So to go through meiosis, we need to divide that in half because you want the baby to have only 46 chromosomes. So if you put an egg with 46 and a sperm with 46 together, you're going to end up with 92 chromosomes, way too many. So you divide that in half, so a sperm only has 23, an egg only has 23. You put those two together, and now you've got 46. So the cell cycle is going to begin just like meiosis does. So interphase is pretty much exactly the same. So the cell is going to grow. It's going to go through DNA replication. So here on the right-hand side here, we're starting, and they're just showing a cell that has 2 different types of chromosomes, a long one and a short one. And they're showing you, again, the red and the blue. Okay, so those are your homologous chromosomes. You got a long blue one and a long red one, a short blue one, short red one, 4 total. Now you go through DNA replication. So this happens in interphase. Remember, S phase of interphase is when DNA replication happens. And now you've got duplicate copies. So here are your sister chromatids, making those X shapes. So you've got 2 copies of each one of those chromosomes you started with. Now here's where the difference lies. For meiosis, you're going to divide those cells in half twice. So dividing the nuclei in half the first time, this is just a schematic, it's not showing you all the steps, but it's just showing you meiosis 1 here divides it in half. So now you have 1 set of the long chromosomes, 1 set of the short in each of those cells. Divide each one of those in half again. You've got 1 copy of the long, 1 copy of the short in each of those cells. So you've got half down here compared to the number you started with up here at the top. The phases are named the same in meiosis as they are in mitosis, but there are a few more steps involved in a few of those stages to keep in mind, especially in prophase. There are extra things that happen in prophase, and they really have divided this up into individual sections here. So just the same as what we talked about before in prophase, the chromosomes are going to condense. So you're going from interphase, where it's a big ball of yarn here, and here the chromosomes have condensed. They've packaged up into these tighter X shapes. And then what's different here is that the homologous chromosomes are going to come together. So you've got chromosome 1 that was from your mom and chromosome 1 that was from your dad, and sister chromatids of each of those. All 4 of those are pairing up together here through this process of synapsis. Crossing over is going to happen here as well. So these are hard to see because they've colored them all the same. They're all purple. But if you see these middle 2 right here in the middle, they've crossed over with each other, and they're going to exchange information. And we'll see that better on a future slide. That's going to help with diversity to make every single one of those gametes different. Additionally, other events that you're used to hearing of, either in prophase or prometaphase, are also happening. So your nuclear envelope is going to fragment, right? It's going to give that spindle access to those chromosomes, and the spindle is beginning to form here. So you've got your microtubules forming to form that spindle. Okay, prophase is even further divided up into these 5 different stages. So we've got leptotene, zygotene, pachytene, diplotene, and diakinesis. They've just really been able to really specifically study each of these events, and they happen at in a very specific order. So they've been able to separate those that phase out into smaller pieces. So your book is calling it a bivalent. We usually call it a tetrad, and that's when you have these 4, here we go, right here in zygotine, if you look here. So these 4 are lining up together. So these, for example, this red one and this blue one that are lining up together, these would both be chromosome 1, for example, or both of them, you know, chromosome 14, but they would be homologous chromosomes, and all 4 of them line up together. A lot of times you'd see that called the tetrad, and you'd see that over here on the left-hand side, that word. Your book calls it a bivalent. Just so you know, those are the same thing. So those are going to pair up with each set of those homologous chromosomes. And you can see here there's a bunch of proteins that help that happen. They're going to help attach all the way along that chromosome to help hold those together. It's kind of like a zipper structure, and it's called the synaptonemal complex. The synapsis is the event that's happening, and it's going to hold those chromosomes together. The next picture we have the crossing over, and here you can see it better than the last slide because now you've got the blue and the red here. Here it's showing you a pretty good picture here. You've got the blue and the red chromosomes because they're homologous, right? So all the same genes are there. Now the alleles might be different, but the same genes are there. They're going to cross over and they're going to exchange some information. So now that piece of red chromosome is going to go with the blue one. This piece of blue chromosome is going to go with that red one. These points at which they cross over are called chiasmata. This word here on the left-hand side, chiasmata. And that's the point at which they cross over. And this can happen many, many times they just show it once here for simplicity, but it can happen many, many times throughout that chromosome on both sides of the centromere. It does require double-strand breaks that happen and repairs through homologous recombination. This, because they're right side by side, they're doing it on purpose. It's not a break from, you know, radiation or something like that. The repair process is much simpler and is much more high fidelity than what we talked about when we were talking about mutations and those repair processes. Okay, so that happens. The synaptonemal complex will kind of start to disappear to help that crossing over occur. And then at the very end, we see our nuclear envelope fragmenting. And now the synaptonemal complex is not really there anymore. You can see there's none of those proteins holding it together. That's going to be important to make sure that they can be pulled apart appropriately. So that synaptonemal complex only lasts through the middle of prophase here to make sure that we can move on to the next part of the process. Okay, here are just some microscope pictures to help you kind of see what it looks like under the scope, because it's a little bit different than what it looks like in a cartoon that's in your textbook. So in leptotene, these are starting to condense, much more condensed in zygotene, and they're going to start to pair up. So you can see these are starting to pair up with each other. In pachytene, it's hard to see here, but crossing over is starting to happen. You can see it much better down here in diplotene. So notice these chromosomes, they're crossing over at least 4 times here. So that, like I said, that can happen many times, especially the longer the chromosome is, the more crossing over you're going to see happening. And then diakinesis, you can see the synaptonemal complex isn't there anymore, so it's not being held together as tightly. So look at this kind of bubble here in between where it's crossed over, and that will get it ready for the next part of the process. Okay, moving on to metaphase. So now, same idea, chromosomes are still lining up here at the center, but all 4 of that tetrad are going to stay together at the center. So that when we move on to anaphase, when they pull apart, homologous chromosomes are going to split. Sister chromatids are going to stay together. So here are your identical copies of sister chromatids here, and they will stay together at this point. In metaphase, it does not matter what order these line up in. So here they're all colored purple. I made these little diagrams down here just to emphasize the point. So we had ours colored red and blue. So for example, for say this is chromosome 1 and these are all lined up together, it doesn't matter if the blue one's on top or if the red one's on top. So it could be like this or like this, either way. And same thing for the next one. It doesn't matter if blue one's on top or red one's on top. So because of that, you might get a complete mix and match of what chromosomes are in each cell, but you will have all of the right types. Just it might be one from your mom or one from your dad. Um, so they get pulled apart in anaphase, and then telophase and cytokinesis, same idea. They're going to chromosomes decondense, nuclear envelope reforms. Okay, now we're moving on to meiosis 2. So we're going to divide these cells once again. Notice two things. One, we are not going through interphase again, so there's no replication of DNA. So you're just going straight from meiosis 1, where you divided, you're just going to do it meiosis all over again. No interphase, no DNA replication. Just do the chromosome splitting part. Here it's showing you that we're beginning with 2 cells because that's what we left off with at the end of meiosis 2. So we had already divided from 1 to 2 cells. Now each of those is going to divide into 2 cells. So when you look at this diagram, the stuff that's happening on top is exactly the same as the stuff that's happening on bottom, just with 2 cells, because that's what you ended with meiosis 1. The result is 4 daughter cells with 1 copy of each chromosome. So notice here you've got prophase 2. This one is much more like mitosis. Okay, so these chromosomes are condensed. You're losing the nuclear envelope again. Metaphase is lining up at the middle, and now your sister chromatids are going to split. Okay, so meiosis 2, the sister chromatids split so that each of these new daughter cells that's going to occur each have 1 copy of each chromosome. So we started with 2 copies of each, and now we have 1 copy of each in each of those gametes. Notice also there's no crossing over in prophase, so none of that extra stuff that happens in prophase 1, none of that happens in prophase 2. All of that's already done with. All we're doing is going through that process of dividing, so this part's very much like mitosis. Okay, so here's just another schematic to show you how these gametes are going to be different. So here we've got chromosome 1, and I'm showing you homologous chromosomes with these kind of darker and lighter shades. Here you got chromosome 2, a darker and lighter shade. So in this case, you know, darker shades from dad, lighter shades from mom, something like that. In this cell, now we've gone through DNA replication, so now, this cell right here. Now we've gone through DNA replication, so we've got sister chromatids. I'm just going to work with chromosome 1 for these next 2 cells just for simplicity. Here you've got crossing over happening. Here's after crossing over, you've got a little bit of that pink one on there, a little bit of that brown one on that chromosome. So you go through meiosis 1, meiosis 2, you split them all apart. What I want you to notice here is look at chromosome 1 in each one of these daughter cells. Each one has a different copy of chromosome 1. This one's got the all brown one. This one's got mostly brown, a little bit of pink. This one's got mostly pink, a little bit of brown. This one's got all pink. Okay, so each one of those got a slightly different version of chromosome 1. So different alleles available. This is important for diversity. Each of those gametes are going to be different. So if you have siblings, this is happening, you know, every time a couple has a baby, you get all these different types of chromosomes. So there's literally, unless you're an identical twin, there's literally no chance that you're ever going to have a sibling that has the exact same combination of chromosomes in their cells from your mom and your dad. Also, what I want you to take note of here is that when we're talking about these homologous chromosomes up here, and I'm kind of distinguishing like, okay, this one's from mom, this one's from dad. Okay, this meiosis 1 and 2 is happening in your cells, for example, to make your eggs or your sperm. These chromosomes you got from your parents. So really, what you're giving your child is some combination of your parents' chromosomes. So they've got kind of a mix match of their grandparents' chromosomes in your egg or your sperm, and then your partner's grandparent or parents' chromosomes in their egg or sperm. So just keep that in mind. Okay, so um, all that diversity is going to lead to diverse populations, and we see that in with human siblings, but usually you only have maybe a couple. So it's easier to look at in something like these puppies, right? You can see that they all have a little bit different markings, right? You can see that diversity coming through in that one litter of puppies. Each of them had a different egg and different sperm, same parents, different egg, different sperm, but different combination of chromosomes that's going to give them a different set of traits. So in biology, when we're talking about diversity, the goal of diversity is survival. Okay, we're talking biology, not sociology, not psychology, just biology. So organisms in nature, the way this benefits them to be diverse is that it's hedging the bet that if something comes along, at least one of these organisms is going to survive and go on to keep the population going so they don't go extinct. So same idea if we're talking with bacteria or if we're talking with, plants that produce thousands and thousands of seeds, right? Each one is slightly different. It's hedging its bets that based on the climate, based on the environment, based on the soil and the nutrients, that one of those seeds is going to make it through and be able to survive. So take a look just at these odds. You don't have to keep in mind this math, but just for an example, we're talking about crossing over, and we were just looking at one or two chromosomes. But you're a human. You've got 23 different types. If you did not have any crossing over, all right, if we're just shuffling, if you got the red copy or the blue copy of chromosome 1, red copy or blue copy of chromosome 2, that would lead to over 8 million different gametes. Okay, so this is the reason why you're never going to have a sibling that has the exact same combination that you have. So 8 million different gametes from those two same parents could occur. Well, actually, that's just from one parent. So 8 million types of eggs from your mom and 8 million types of sperm from your dad. Those are your odds. If you had one crossover event per chromosome, okay, and we know that I already told you that it's usually more, especially on those bigger chromosomes. If you just had one crossover per chromosome, now we're up to 4.9 times 10 to the 27. Okay, so that's just an outrageous number. So there's just virtually no way other than identical twins that you can get to people from the same parents that are exactly the same. Okay, so when we're talking about these homologous chromosomes, what exactly makes them different? So why is chromosome 1 from your dad different than chromosome 1 from your mom? Now, all the same genes are there. So if on chromosome 1 there's a gene for a certain enzyme that has a certain job, there's a gene, for your hair color, for example. I'm just giving you some examples. But you've got all those same genes for the same proteins. However, there are alleles of these genes, so different versions of the same gene. You can kind of think about it like different flavors of ice cream. Okay, it's all ice cream, but they're different versions for the same one. For example, what it's showing you here is here's blue chromosome, red chromosome from two different flowers that they're mating together. In the blue chromosome, the gene for flower color has a C at this position. In the red chromosome, there's an A at that position. Okay, these are not always called mutations. Sometimes it probably resulted from that originally, but it's really not going to affect the plant other than if you've got this copy, it's going to give you purple flowers. If you've got this copy, it's going to give you white flowers. That's indicated by what they've told us here. So they've got different flavors of that gene, okay? It's not going to hurt the plant, just going to give it a different trait. The genotype is the actual genetic makeup of an organism. So what alleles do you have? So it might be saying that you have this particular sequence. A lot of times we use just letters to denote these. So this might be the big P for purple, and this might be a little p down here for the white ones. but that's telling you what copies of the gene do you have. If you have two of the same one, so if in this case you actually had, if both of these chromosomes had this one for the purple flowers, we would call it homozygous. Homo means same. If there were two different ones, like it was shown here, so you got this chromosome has a purple, this chromosome has a white, we call that heterozygous, so hetero for different. When it comes to how these are expressed, so this is just the DNA we're talking about here. Remember, it goes through the process of being made to RNA and then made to protein, which is going to make some sort of enzyme that does a job. In this case, that enzyme synthesizes that purple pigment to give the flower a purple color over here. One copy is usually dominant over the other. So usually you see one or the other. In this case, this one produces an enzyme that makes a purple pigment. If you have this copy of the gene, that protein doesn't work and doesn't hurt the flower, but you just don't have that enzyme. It doesn't make that pigment, so your flowers are white instead of purple. So in this case, the purple is the dominant one, and most of the time that's completely determined by experimentation, right? Just figuring out if I make these two together and I have both of these present, which one do I see? That would make the other one recessive. It's just kind of hidden. You've got the allele for a white flower, you're just not seeing it. All the flowers here are purple. And then the phenotype, remember, is the physical trait that you see. And I put C in quotation marks here because sometimes it's on the outside. So for example, purple pigment in a purple flower. But sometimes it's something else. So say you're studying how somebody digests a certain molecule. You can't see that necessarily in the outside, but you could take a blood sample or a urine sample or something like that, and you can measure it. So it would be the physical trait still, but you might just not see it with your human eyes on the outside. You might have to measure something else with that person. So this is how these homologous chromosomes, right? Still both chromosome 1, for example, both have the gene for flower color that's going to indicate flower color, but they just have different versions of that same gene. Okay, this takes us into some of Mendel's laws for inheritance. We're just going to go through these briefly. So remember, Mendel was a monk. He did all those experiments with pea plants. We talked about that in 1401 to determine how are these things inherited. And at that point, he had no idea about DNA. Only that came, 150 years later after he made his observations. But what he observed and they are still true today. Phenotypic traits, so what you see, the physical traits, those are determined by what genes you have, what alleles of what genes. And again, at that point, he called it something different because they didn't know what DNA was, they didn't know what genes were. That terminology came later. But any trait that you have, whether it's your height, your hair color, how you metabolize something, all of that is determined by what genes that you have. Number 2, organisms inherit 2 alleles of a gene, right? You got 2 copies of everything, one from your mom, one from your dad. How those interact is going to give you what phenotype you have. The law of segregation, the 2 alleles of a gene segregate during meiosis, meaning one egg or sperm only gets 1 copy of each gene. And that's true because you only get 1 copy of each chromosome in that gamete. And then the law of independent assortment is that the alleles segregate independently from alleles of other genes. Meaning if I'm looking at the alleles for hair color and then I'm looking at the alleles for height, you could have any combination. So if your dad was really tall and had brown hair and your mom's short with blonde hair, you could be tall with blonde hair, you could be short with blonde hair, you could be tall with brown hair, you could be short with brown hair. So they aren't necessarily linked. Now, I put a caveat here that this only applies if they're on different chromosomes. So if one trait is on chromosome 1 and the other trait's on chromosome 2, you can get any combination of those two. If they're on the same chromosome, then they're more likely to be inherited together, and that's something we would go through in genetics class. Okay, if meiosis doesn't occur correctly, you would have something called non-disjunction. So we talked about this a little bit when we were talking about mitosis and what would happen if the spindle didn't attach correctly. So, for example, you can see up here the cells, going through anaphase. And remember, we talked about if the spindle doesn't attach on one side, then all of those get pulled to this side, and this side gets none of that chromosome. So if you go follow this cell on down, now you've got this cell that's got extra chromosomes, this cell that's missing one, and at the end of the day, these two gametes have an extra chromosome, these two gametes are missing a chromosome. So that's if this non-disjunction event happens in meiosis 1. It can also happen in meiosis 2. So here, meiosis 1 happened just fine. The homologous chromosomes separated. Now we're in meiosis 2 down here. Now's our part where the microtubules didn't attach appropriately, and both of these chromosomes went to the same cell. Now this cell has one too many, this cell has one too few, but these other two cells are fine. So these have the exact numbers they should have. They're half of what we started with, and they will work out fine if they're combined with another gamete. So it's more severe, obviously, if it happens in meiosis 1 because all 4 of these are screwed up, versus if it happens in meiosis 2 where you've got 2 out of 4 which are just fine. This tends to happen more with the smaller chromosomes than the large ones. Remember back when we were talking about DNA, I showed you that karyotype that had all the chromosomes on it. They're numbered 1 through 22, where 1 is the biggest one and 22 is the smallest one, and then you've got X and Y. So the higher numbers of chromosome are the smaller ones, and this tends to happen more easily with those. It's harder for them to get attached to the spindle than the larger ones. And like I mentioned here, it can result in too many chromosomes, like in these cells, or too few. Good example of this, Down syndrome, trisomy 21, that's when you have 3 copies of chromosome 21. So, for example, if this is an egg and then it joined with a sperm that had 1 copy of chromosome 21, and this one has 2 copies of chromosome 21, now you got 3 in that baby. Okay, this is a karyotype picture like I just mentioned. So here you see the largest chromosome, which we call chromosome 1, on down to the smallest one, chromosome 22. This is a karyotype of a person with Down syndrome, so they have 3 of chromosome 21. And this, obviously, this karyotype is done during mitosis. So they probably they might have used cheek cells or blood cells. I'm not really sure for this. So they've gotten it when they're replicating. So you can see the little X shapes formed of the sister chromatids with all these chromosomes. Anytime you have an abnormal number of chromosomes, we call that aneuploidy. And you can get more specific than that, but just as a general term. So this word here, not diploid, not haploid, but aneuploidy meaning an abnormal number. Down syndrome is the most common. That's trisomy 21, but there are a few others that are also common, also down here in the smaller chromosomes. All the others, you don't really see them in a baby or in a person because they're too severe. So they will be lethal way before birth, usually in the first trimester. So most miscarriages, maybe not most, I would say the statistic is somewhere between 30 to 50% of miscarriages in the first trimester are because of chromosome numbers not being correct. So they're too severe, the baby will not develop, the embryo will not develop. And remember also, I told you animals just don't do well with having extra copies of chromosomes. Much easier for plants, but animals can't handle it through their developmental processes. The reason we do see more of Down syndrome is because this one's much less severe. Like a person can live and have a pretty full and fairly normal life if they've got an extra copy of this teeny tiny little chromosome 21. Not as many genes on there, not as detrimental to the person. Okay, and then just a quick note that we've been talking about the gametes being formed as if they're all formed in the exact same way, and it's not quite true. So I just wanted to just bring that up. So with sperm, it's pretty much exactly like we talked about, right? So going through meiosis one, splitting apart, splitting a second time, and you end up with 4 sperm cells that all have 1 copy of each chromosome. With females, it is not quite the same. So here it shows you the starting cell, and see how during metaphase, it's kind of way off to the side here. It's not like in the middle like it is with this cell up here. And you actually have it pulled apart, and it's not equally divided. So when you go through cytokinesis and telophase, it does not divide in half like we're used to seeing. It actually makes this teeny little cell, which we call a polar body. This one will not be used for a mature egg. And it's going to go through the process of meiosis 2 and split those again. What you end up having is one big cell. This one's the egg cell, and it will go on to be a mature egg, and it's the one that can be fertilized by a sperm. These 3 are polar bodies. Those are going to get reabsorbed. Those chromosomes aren't going to be used. Nothing's ever going to happen with these. They're just going to kind of disintegrate and dissolve. So those polar bodies don't get used at all. So in effect, you go through meiosis and really only make 1 egg cell fully. Now, another fun fact is when you are born, all females are born with all the eggs that they will ever have. Your eggs have already been made when you're a baby. But they're frozen in meiosis 1. So they're kind of here in this stage, right in here, until you hit puberty. Now, once you hit puberty and you start menstruating, that's when every month, once a month, it will continue through meiosis 2 to produce one of these egg cells that will be ovulated. And actually, it doesn't completely finish meiosis 2 until that egg cell is fertilized. So it's just a little bit different process. The timing is very different, whereas the sperm cells are, every time they get made, it's happening pretty much right at that time, whereas meiosis for females is really spread out and happens a little bit differently.